Hello and welcome to this AS level biology tutorial on diversity and classification. The aims of this tutorial are to define what phylogeny and taxonomy is, list the differences between phylogeny and taxonomy, define species and explain the traits organisms of same species share, define genotype and phenotype, list the causes of different phenotypes, list and explain briefly the ways of measuring genetic diversity, remember common terminology used in ecology, and finally define biodiversity, how can it be measured. So there are a lot of points to cover, so we'll get right into it. So organisms can be classified in two ways. One way of classification is phylogeny. Phylos means race and geny means origin. So phylogeny, literally put, shows the origin of all organisms and their common ancestor. The specification describes this as the study of evolutionary history and relationships between different species of organisms. Now let's look at a phylogenetic tree. Here, the halophiles and Methanosarcina, sorry about my pronunciation, are more closely related than Methanobacterium, seen here. This is because closer the branches are, the more closely related the organisms, and the more recent common ancestor they share. I hope that made sense. Another way of classifying organisms is via taxonomy or hierarchy. These terms are almost exactly the same, and the word taxo means arrangement and nomi means law. Here, the organisms are arranged according to a set of rules. The specification defines this hierarchy/taxonomy as a smaller groups placed within larger groups with no overlap between them. Let's look at this image of a hierarchy. Note that as you go down, the organisms are more similar and decrease in numbers, and vice versa if you go up. These kingdom, phylum, class, order, family are all called taxon. A good way of remembering it is Dr. Kevin Peterson coughs green sputum. Of course, if you don't like this mnemonic, you can make your own. Now let's look at how organisms are named according to their taxonomy. Organisms' names are usually written in the binomial format, the bi referring to the two. The first part is genus, and the second is the species. Note that both are written in italics and the genus has a capital letter. Taxonomy allows for universal communication of an organism, such as the name Nymphicus hollandicus, also known as a cockatiel. And taxonomy names are useful because they never change, while local names may do. The word cockatiel may be more familiar to the UK, whereas it would be a different term in some other country. Now let's summarize the differences between taxonomy and phylogeny. Taxonomy arranges organisms to a set of rules, while phylogeny doesn't. Taxonomy aims to name and classify organisms, while phylogeny aims to show the evolutionary relationships. Taxonomy does not show the shared evolutionary history of an organism, while phylogeny does. I hope you've understood it now. Now let's move on to defining species. Species are a group of organisms that can interbreed to produce a fertile offspring. Organisms of the same species have similar courtship behavior. I say similar because if they had the same behavior, animal romance would be quite boring. They have similar phenotype but same anatomy. That is to say, animals of the same species will have the same body structure but may differ in color, height, etc. Finally, organisms of the same species have same genes but different alleles, hence why their phenotype differs. A point to note is that organisms that look very similar externally may not necessarily belong to the same species. This is important because organisms like the cockatiel and the cockatoo look very similar, but they are completely different species. However, it does suggest that they shared a more recent common ancestor. Now let's move on to the terms genotype and phenotype. Genotype is defined as all the genes that make up the organism. It's inherited. Whereas phenotype are the observable characteristics that is a result of the effect of the genotype and the environment. Causes of different phenotypes include environmental selection pressures, gene mutations which produces new alleles, meiosis which causes different combination of alleles, and the random fusion of gametes, again causing the different combination of alleles. Let's look at how we can measure this diversity, more specifically the genetic diversity. Genetic diversity is described as the variation of alleles in a species of organism. And to determine how similar organisms are, we can do genome sequencing, which can be done via polymerase chain reaction, abbreviated as PCR, or DNA hybridization. PCR is covered at A level, so we'll not cover it in this video. Other alternatives are amino acid sequencing and immunological comparisons. Now let's understand the process of DNA hybridization. As you may know, the word hybrid involves two different things, such as hybrid cars, which have an electric and a petrol engine. And it is very similar to the hybridized DNA, which is made in this process, and it contains DNA of two different organisms. The process involves one organism's DNA being radioactively labeled. Then the DNA is heated to break the hydrogen bonds between the strands. And as the strands separate, it's cooled so that the single strands of different organisms can join, and this we call as hybridization. We can identify the hybrid DNA by measuring the radioactivity, or what we could do is heat the hybrid DNA to determine the temperature the strands break. Overall, what the results should show is that greater the temperature equals more similar the DNA sequence and more similar the organism. I hope this image makes more sense. As you can see, the organism's A DNA is hybridized to organism's B DNA. And if it has complete binding of the DNA bases, that means that the organisms are genetically identical. If there is partial hybridization, so that not all bases join to the DNA, to the template strand, then the organisms are related. And if there is no hybridization, the organisms are completely unrelated and they don't have a recent common ancestor. Let's move on to amino acid sequencing. It involves the comparing amino acid sequence of the same protein. The more closely related organisms are, the more similar amino acid sequence they have. If they have a similar amino acid sequence, they have a similar DNA base sequence, hence meaning that they share a recent common ancestor. I hope this diagram makes more sense. You can see that the base sequence of human is identical to the chimpanzee, and only one amino acid difference to the gorilla. This means that we are more closely related to the chimpanzee than the gorilla. However, there is a problem with amino acid sequencing. It's because DNA is degenerate, which means the amino acid could be coded by different base sequences. Finally, we can do immunological comparisons. This involves injecting specific shaped antibodies to target specific shaped antigens, example the human antigen. The antibody then binds to the antigen to form an antigen-antibody complex, that is, a precipitate. More complementary the protein to the antibody, more precipitate is made, and more closely related the organism. However, be careful, as this is only one type of method and examiners can adapt this differently, so read the question carefully. This diagram on the right explains why. Here, human serum, which contains human antigens, are injected to the rabbit. The rabbit generates an immune response creating antibodies for the human antigen. This anti-human antibody is then injected to different organisms to compare the amount of precipitate made. The gorilla forms the most precipitate of all other animals, meaning it has an antigen that has a very similar shape to the human antigen. Now we move on to AS part of ecology. Boo! I know, it wasn't my favorite topic either. The terminology you need to know here are biodiversity, which is the variety of living organisms in an area, habitat, which is the location where organisms live, community, is where all the population of different species in a habitat, species richness, the number of different species in a community, intraspecific variation, which is variation within organisms of same species, interspecific variation, which is variation of organisms between different species. Note that a low biodiversity is associated with low genetic diversity. Remember, biodiversity and genetic diversity are not the same terms. So how can we measure this biodiversity? Well, there are two ways. One is species richness, which is the number of species in that habitat. Or we can do index of diversity. The index of diversity has a formula, and this is it. You don't have to remember this, but you do have to use it. The capital N stands for the total number of organisms of all species, and the little n stands for the total number of organisms of one species. And the D stands for biodiversity, not genetic diversity. Higher the value of D, greater the biodiversity. If D equals 1, then the organisms are of the same species. I've done an example for you so you can understand what's happening. So the question says there are 52 birds consisting of 5 different species. Calculate the biodiversity. We can form a table consisting of 5 different species, A, B, C, D, E, and they will be given in the exam, don't worry. And then we can label little n, which is the number of organisms of each species. Then calculate n minus 1. Again, I'm sure this makes sense on the table. And then we multiply n into n minus 1. Once we have all done this, we have to do the sum. That's what this sigma symbol says. So the sum of all this, and this equals 522. Remember, capital N is the total number of organisms of all species, and the total number of organisms of all species is 52. So we do 52 times by 51 divided by 522, which equates to a diversity value of 5.08. Again, I hope this made sense. Alright, nearly done now. The last thing I want to talk about is the effect of agriculture on biodiversity. 3 main things that agriculture has is that deforestation destroys habitat, causing some organisms to migrate or die. Planting one type of crop reduces the types of species that can feed off it. And the use of pesticides and herbicides, which kill animals and plants respectively, reduces their diversity. This effect can be lessened by conservation schemes to help protect the habitat and maintain diversity. Examples are using less herbicides and pesticides, and nature reserve parks. Again, these are fairly common sense, and most of this shouldn't be a surprise. That's the end of this tutorial. I hope you understood diversity and classification. Here's a recap of the aims that we went through. Use the aims as questions to strengthen your knowledge. Thank you.